As we talk about the derivatives of the ectoderm and the neural crest cells, we are once again looking at the dorsal view of the embryonic disc. We have seen that there was a formation of primitive streak from the caudal to cephalic end, and then the epiblast cells were migrating. On the ventral side, there was endoderm. In the middle, there is a mesoderm. Whereas the epiblast cells, which are remaining dorsal only, they'll form the ectoderm. Now, there will be four types of ectoderm which we have to focus upon. One which is central is called as the neural ectoderm, which will then form the neural plate, neural groove, and neural tube, the central nervous system. So, near the midline is the neuroectoderm, whereas the periphery will have the surface ectoderm. Then there are two more which we have to discuss. Let us first look at the details here. We can mention that this is the cranial end, where you will find there is a membrane. We are calling it as buccopharyngeal membrane. And then this is the caudal end, where we have mentioned the cloacal membrane. And then this is the central area. Near the midline, we are having the neuroectoderm. Now, if that is the neuroectoderm, what lies at the periphery? At the periphery, we have surface ectoderm, which we can mention here. Now, once we have talked about the surface ectoderm as well, as mentioned here, we find that near the neural ectoderm, there are some neural crest cells coming. And these neural crest cells, some of the authors will mention they come from neuroectoderm. But we have learned earlier that in the latest embryology books, these neural crest cells are considered to be the fourth germ layer coming from the epiblast cells. So let us mark them here. The neural crest cells close to the neuroectoderm, and then there is another entity which we are calling as the placoidal ectoderm. And what is the function of these placoidal ectoderm? It is actually going to give us contribution to placoids like the nasal placoid for olfactory purpose, otic placoid for the hearing and balance. Then there is optic placoid. So as we can understand, ectoderm is divided into four components. The central portion is the neuroectoderm. Outside that are the neural crest cells. Still in the periphery is the placoidal ectoderm, and the most peripheral is the surface ectoderm. And we are going to make this diagram ourselves as well. But our diagram will be our transverse section, which we are taking at this level, and then we can talk about different type of ectoderms and their future derivatives. Our diagram will be something like this: a transverse section of the embryo showing a dorsal amniotic cavity, ventral yolk sac cavity, and the three germ layers. We are going to focus upon the ectoderm, which was dividing itself into various components. This axial mesoderm, the notochord, is stimulating the overlying ectoderm to form neuroectoderm, whereas in the periphery will be the surface ectoderm. So let us draw this diagram ourselves now. Here we are drawing the transverse section of the embryo, and on the dorsal side we have amniotic cavity, on the ventral we have the yolk sac cavity, and after gastrulation we have the three germ layers formed. The dorsal is the ectoderm, which we are going to focus upon. In the middle we have the mesoderm, and ventrally we have the endoderm. Then we have mentioned that there was one axial mesoderm here, and we were actually calling it as the notochord. So this notochord or the axial mesoderm is important for us because it is the one which is going to stimulate overlying ectoderm to convert into neuroectoderm, and we can mention it here then. The central portion of the ectoderm is going to convert itself into the neuroectoderm, whereas if we are looking at the periphery, maybe on the right side or on the left side, this is going to be the surface ectoderm. Now, before we start talking about these two types, we know there were formation of neural crest cells as well. And these neural crest cells, as we are telling, some of the authors are mentioning the neural crest cells are derived from the neuroectoderm here. But recently we have realized these neural crest cells are actually the fourth germ layer, and they are coming from the epiblast cells. They are contributing to the peripheral nervous system along with some other derivatives. Like in the PNS, they are going to form the ganglia. Most of the ganglia are derived from the neural crest cells. Then who is going to form the CNS? The central nervous system is developing from here, the neuroectoderm. One plate will form, the neural plate ectoderm, and then this neural plate will become the neural groove, will become the neural tube. It is this neural tube which is going to form the central nervous system, which is basically basically the brain and the spinal cord. What else these neural crest cells are going to give us? The neural crest cells have several derivatives, like they are going to form secondary mesenchyme, which is mostly restricted to the anterolateral aspect of the head and neck region. There are two types of mesenchyme, primary and secondary. The secondary mesenchyme, which may come from the neural crest cells, restrict itself to the anterolateral aspect of the neck region, doesn't go posterior. Posteriorly you have primary mesenchyme, and rest of the body has primary mesenchyme. Then where is this primary mesenchyme coming from? As we mentioned here, the neural crest cells can give us the secondary mesenchyme. By the way, what do you understand by mesenchyme? Mesenchyme is basically embryonic connective tissue, and some of the books will call it as mesoderm, though they are not interchangeable term, but for most of the purpose, you can call it as mesoderm. So the secondary mesenchyme is coming from neural crest cell. Who is forming primary mesenchyme? If you remember, I have mentioned this middle layer, which most of the books are calling it as mesoderm, is written as mesoblast by Gray's Anatomy. So here, this middle layer, which is called as mesoblast by Gray's Anatomy and mesoderm by some of the other authors, is the one which is going to form primary mesenchyme. So there are two types of mesenchyme. Most of the body is having primary mesenchyme. Primary mesenchyme is present on the posterior aspect of the skull as well, whereas the secondary mesenchyme is restricted to the anterolateral aspect of the head and neck region. Like most of the skull bones, including facial skeleton, is from secondary mesenchyme. Most of the eyeball mesoderm is secondary mesenchyme. The pharyngeal arch bones like mandible bone, hyoid bone, they are from the secondary mesenchyme. So we can mention here, like most of the skull bones, especially anterolateral, and most of the eyeball, especially the mesoderm, and most of the pharyngeal arches are from the secondary mesenchyme. So it means there'll be some exceptions, because you're mentioning most of the, yes, like most of the skull bones, but which are anterior and lateral. If you are talking about occipital bone, it is not from the secondary mesenchyme. It is not from the neural crest cell. It is from the primary mesenchyme. So you can mention primary mesenchyme is going to form the occipital bone in the skull. Then as we mentioned, the pharyngeal arches, mostly they are coming from the secondary mesenchyme, the neural crest cell. But remember, neural crest cell can form pharyngeal arch bones like mandible bone, the hyoid bone, but not the pharyngeal arch muscles. Neural crest cells do not form the pharyngeal arch muscles. They are skeletal muscles, and all skeletal muscle must come from the mesoderm. So it is the mesoderm which is going to form primary mesenchyme and pharyngeal arch muscles. You can take one example like masseter muscle. So you mention here the primary mesenchyme is going to form pharyngeal arch muscles. For example, the masseter muscle. So the point is, neural crest cell can form pharyngeal arch bones, but not the pharyngeal arch muscles. The muscles are from primary mesenchyme and bones are from secondary mesenchyme. Then you can mention neural crest cell or the secondary mesenchyme forms the aorta pulmonary septum as well. And we can have aorta pulmonary septum anomalies like transposition of great vessels in conditions like DiGeorge syndrome. DiGeorge syndrome is a problem of neural crest cells, and in DiGeorge syndrome, which happens due to problem of the migration of neural crest cells, we find that the baby is having a problem of the skull bones. There will be skull bone defects, eyeball defects, pharyngeal arch defects, a small mandible hyponatremia, and episeptum anomalies like transposition of great vessels. In fact, the most common cause of death in DiGeorge syndrome is aorta pulmonary septum anomalies. Now let us continue with some more derivatives of the neural crest cell in our diagram. As we mentioned, neural crest cell is contributing to peripheral nervous system, and most of the ganglia are coming from neural crest cells. As you will notice, maybe it is some cranial nerve ganglia like cranial number 5, 7, 9, 10, or the ganglia which are related with the spinal cord like dorsal root ganglion, the sympathetic chain ganglion. Their neuron bodies are coming from neural crest cells. The neural crest cells are migratory cells and they are migrating throughout the body. Like you see here, they have reached in front of the aorta, in front of the vertebra. The prevertebral ganglia are contributed by neural crest cell. Like you can take one example, celiac ganglion in relation to the celiac artery is coming from from the neural crest cell, controlling the foregut derivative. Even in the gut, the enteric ganglia, Arbex and Meissner ganglia, which you can see here, they're also coming from neural crest cell. So may it be the Arbex ganglia or the Meissner's ganglia, they are coming from neural crest cells under the category enteric ganglia. And in disease like Hirschsprung disease, what happens is the neural crest cells are not migrating and reaching the distal colon. And obviously, if you do not have neural crest cell, you will not have the ganglia, and there'll be a ganglionic colon, leading to some retention of fecal matter, enlargement of the colon, megacolon. We'll be discussing all those details, but the basic problem is non-migration of neural crest cell, absence of neural crest cells, absence of the ganglia, and these neural crest cells are also reaching adrenal gland, the adrenal medulla. There are some sympathetic neurons inside the adrenal medulla, and those sympathetic neurons are coming from neural crest cells. Let us look at one more diagram where we can see some more derivatives of neural crest cells. We mentioned that the neural crest cells are going to form secondary mesenchyme, which is mostly restricted to the anterolateral aspect of the head neck region, forming most of the skull bones, most of the eyeball mesoderm, and which you can see here. In the eyeball, most of the mesoderm is secondary mesoderm. But then you can mention in the eyeball there are some contribution from the surface ectoderm as well. Say for example, first layer of cornea and the eye lens. If you are looking at the first layer of cornea and the eye lens, they are actually from the surface ectoderm. Then there will be some derivative of the neuroectoderm as well. We will see the developing eye in later discussions, but at the moment, the neural crest cells forming secondary mesenchyme contribute to most of the skull bones as well, like the facial skeleton, but not the posterior bones like occipital bone, because occipital bone is coming from primary mesoderm. So you can mention this is going to be an exception because it is coming from primary mesenchyme, not the secondary mesenchyme. The neural crest cells were migrating to the cardiac region, forming the aorta pulmonary septum, which is also called conotruncal septum. We have mentioned in DiGeorge syndrome, there is a problem with the aorta pulmonary septum, leading to some anomalies. And these neural crest cells, they were forming the pharyngeal arch bones as well, like they form malleus incustapes, developing in first and second pharyngeal arch, also forming the mandible bone, the hyoid bone. But then we mentioned the neural crest cell forms secondary mesenchyme, form the pharyngeal arch bones, but not the muscles. The pharyngeal arch muscles are skeletal muscles, and neural crest cells do not form skeletal muscle. And where are the pharyngeal arch muscles coming from? They come from the primary mesenchyme, like the masseter muscle is from primary mesenchyme. And then we can mention the skin dermis in the head and neck region is also from neural crest cell. In the skin, if you look at the epidermis, it is from surface ectoderm, but the dermis in the head and neck region, especially anterolateral, it is from neural crest cell. And it is the neural crest cell who will form odontoblast and dentine of the teeth. So odontoblast form the donto or the dentine of the teeth, but then not the enamel. If you're looking at the enamel, then the enamel is coming from ameloblast, and ameloblast themselves are from the surface ectoderm. So in the teeth, on the surface, you have surface ectoderm forming the enamel by the formation of ameloblast. But the teeth proper, the dentine of the teeth, the odontoblasts are from the neural crest cells. So here we have seen some of the derivatives from neural crest cells. What were the exceptions? There were some derivatives from the surface ectoderm. Let us now focus on the derivatives of surface ectoderm. Can you enumerate them here? As we mentioned, surface ectoderm is going to form first layer of cornea and the eye lens. The surface ectoderm is also forming ameloblast, the enamel, and surface ectoderm also forms the skin epidermis. Let us enumerate them in our diagram now. The derivatives of surface ectoderm. And as we come back to this diagram, we have mentioned that the ectoderm, which is at the periphery, maybe right side or left side, is the surface ectoderm. And we want to see what are the derivatives of this surface ectoderm. There's a rule which we have to follow. Any external opening in our body is lined by surface ectoderm. Like the exterior of the eyeball, there is surface ectoderm. At the opening of the nose, opening of the mouth, opening of the ear. So any external opening in our body will be lined by surface ectoderm. You can also mention the openings in the lower body, like in the perineum, opening of the urethra, opening of the vagina, opening of the anal canal. They are all lined by surface ectoderm. So you mention any external opening is lined by the surface ectoderm. That is a rule. Like we have mentioned, it could be the eye, the nose, the mouth, the ear, even the openings in the perineum, which are urethra, the vagina, or the anal canal. In fact, you can mention proctoderm for anal canal and stomoderm for the oral cavity. So may it be the stomoderm or the proctoderm. They are the opening of the gut tube. You are putting your food in the stomoderm and the fecal matter is coming out of the gut tube at the proctoderm. But both of them are lined by surface ectoderm. And then in the stomoderm, at the roof of the oral cavity, one pouch is developing, the Rathke's pouch. And that Rathke's pouch is contributing to pituitary gland, the anterior pituitary or the adenohypophysis. So as we mentioned, it is the surface ectoderm at the roof of oral cavity giving us the Rathke's pouch. And this Rathke's pouch, the surface ectoderm, is forming the anterior pituitary or the adenohypophysis. And what about the posterior pituitary or the neurohypophysis? As the term is mentioning, the other part of pituitary, the posterior pituitary, which is also called neurohypophysis, must be coming from neural ectoderm. In fact, it is the neural plate ectoderm which is contributing to the central nervous system. And in the central nervous system, there will be diencephalon. And from the diencephalon, there is a downward prolongation which is contributing to the neurohypophysis. You can mention here, in the brain, we are having the diencephalon, and it is the downward prolongation of the diencephalon which will form the posterior pituitary or neurohypophysis. So pituitary gland is having contribution not only from the surface ectoderm but also from the neuroectoderm. Both type of ectoderms are contributing to pituitary gland. Let us continue our discussion of the surface ectoderm. It is going to form the surface epithelium, the skin epithelium, skin epidermis, and also the glands there, sweat gland, sebaceous gland. So as we mentioned, surface ectoderm is going to form skin epithelium, which is the epidermis, plus its glands also, like the sweat gland and sebaceous glands. And these glands, they have some myoepitheliocytes to squeeze the secretion out. Those myoepitheliocytes are also from the surface ectoderm. All this information is important because we know they are the questions in our exam. So myoepitheliocytes, which squeeze the secretion of these glands out, they are also from surface ectoderm, which is a strange finding because these are muscles, the smooth muscles, and even the muscles are coming from ectoderm. See, normally muscles are coming from mesoderm, but there are some muscles which come from ectoderm. When we have seen surface ectoderm forming myoepitheliocytes, and we should know that even the neural ectoderm, the neural plate ectoderm, will give some muscles, like the muscles of the iris, sphincter pupillae, dilator pupillae. They come from neural plate ectoderm. You can mention their name here. It is the neural plate ectoderm which will form the iris muscles, like sphincter pupillae and dilator pupillae. Though it is very strange to find the smooth muscles coming from ectoderm. As we mentioned, the oral cavity is the stomoderm and initially it is lined by the surface ectoderm. Even the glands which give secretion in the oral cavity, like the parotid salivary gland or the sublingual salivary gland, submandibular salivary gland, these three salivary glands, they are also coming from surface ectoderm, lining the oral cavity. Actually, we have got a question upon that. Let us look at the question then. This question is asking us about the derivatives of ectoderm and the exception. As we have mentioned, in the skin epithelium, epidermis, is coming from surface ectoderm. So this cannot be the exception. Even the salivary glands, may it be the parotid salivary gland or sublingual submandibular salivary gland, they are coming from the surface ectoderm lining the oral cavity, the stomoderm. So even that could not be the answer. Then what about the neurohypophysis? It is the posterior pituitary coming from neural plate ectoderm. We have known the neural plate ectoderm will form the neural tube, will form central nervous system, the brain and spinal cord. In the brain there is diencephalon, and diencephalon gives a downward extension to form posterior pituitary or neurohypophysis. So it is again from the ectoderm, neural plate ectoderm. That means the only possible answer is choice number D. Then if this is the answer, what is this arrector pylorem and where is it derived from? Arrector pylorem is a smooth muscle in the skin for erecting the hair, and it is coming from mesenchyme, the mesoderm. Be careful that this muscle in the skin is from mesoderm, but then we have seen some muscles in the skin which were from the surface ectoderm, the myoepitheliocytes, which will squeeze the secretion of sweat gland and sebaceous gland. Those myoepitheliocytes, the smooth muscles, they are from surface ectoderm, but in the skin you have another smooth muscle which is arrector pylorem, and that is from mesoderm. So the answer of exception is choice number D here, the arrector pylorem for erection of the skin hair.